Imagine you are suffering from a severe chronic disease. You rely on antibody therapy. However, this therapy, your lifeline, comes with severe adverse side effects. Inflammation, unrelenting headaches, and constant fatigue, just to name a few. Furthermore, its effectiveness diminishes over time. Is this the therapy you would want for yourself? Yet, this is the harsh reality for many patients who rely on antibody therapy, like the student Alexander, who kindly shared his experience with us. But a new revolutionary approach is that to overcome all these problems, B-cell therapy, a therapy applied as a one-time treatment which will constantly produce antibodies in the patient at the right place, at the right time. So we as iGEM Team Munich, we believe in the vision of B-cells as living and evolving drugs. But what exactly is B-cell therapy? The idea is to take a patient's B-cells out of the body, then manipulate them genetically to introduce a specific therapeutic antibody sequence into the B-cell, and then reintroduce this B-cell into the patient's lymph nodes. So this way we can seamlessly integrate known antibody sequences into the endogenous locus of the B-cell, which provides major advantages. So which advantages are these exactly, so compared to standard antibody therapy? So on one hand, what we already said, it's basically a single dose treatment in comparison to regular injections. Moreover, our B-cells are able to form a memory, so this means lifelong protection. Furthermore, we really preserve the natural strengths of our B-cells by enabling them to undergo class switching and somatic hypermutation. So you can really take advantages out of these natural abilities of our B-cells. This therapy, or at least the underlying research, was already conducted by Adi Barzel and his group, who we consulted with extensively to make this project a reality. Our project B-cell engineering and enhancement, is focusing on enhancing this existing approach of B-cell engineering and make it even more efficient. So on which goals did we exactly focus in our project? So we wanted first to expand our target range to 4 different targets to prove its versatility. Second, we wanted to introduce extensively engineered antibodies into the B-cells for the first time. And third, we wanted to increase the safety by implementing additional safety features into our cells. To make this a reality, we consulted with over 20 experts from various fields to implement our B-cells for a variety of diseases. So, consulting with our experts, which targets did we actually choose for B-cell therapy? So, just imagine, with B-cell therapy, we could revolutionize cancer treatment. The major advantage is also discussed with Uwe Platzbecker, who was the first one to implement CAR T-cell therapy in Europe's clinics. He really highlighted this ability of B-cells to form a memory. So this lifelong protection that is offered by our therapeutic approach. So our idea is basically to introduce cancer-targeting antibodies into the B-cell, which are also connected to IL-2, which can be released in the specific tumor microenvironment. So on one hand, we fight cancer with the specific antibodies, which attract other immune cells. On the other hand, with IL-2, we can kickstart a potent T-cell response, just like interlocking wheels. We also focused on targeting acute bacterial infections. So let's just imagine we have a patient who could die if infected with multi-resistant or antibiotics-resistant disorders. But with our B-cells, we could implement a prophylactic approach, protecting him from being infected from this disease. So we basically designed antibodies that were targeting the S. aureus exotoxin A, and thereby prevented the infection and hemolysis of blood cells. So I just said prophylactic approaches. Another prevention method that we want to target with our B-cell therapy are neurodegenerative diseases, because prevention is the key when it comes to fighting neurodegenerative diseases. As also discussed with our expert Oliver Witz, Alzheimer is usually diagnosed several years after onset, and this makes treatment almost impossible. So, but with our approach, we could insert a B cell into a patient, let's say, who is already genetically predisposed to developing Alzheimer at some point, and we could inject this B cell producing antibodies directly targeting the amyloid plaques that form in Alzheimer's disease, which are also able to cross the blood-brain barrier by additional transcytosis mediating blood-brain barrier shuttles that we introduced in our antibodies. So, but with this, we could, of course, not only target neurodegenerative diseases, but we can basically focus on all brain-associated diseases using our transport shuttle. When it comes to these natural strengths that are already highlighted from our B-cell therapy, the perfect target are chronic viral infections, such as hepatitis B virus, because our B cells are still able to undergo class switching and somatic hypermutation. So our B cells are able to quickly react to these quickly mutating viruses and thereby protect a patient for his life.
Well, now we already talked about extensively about the various advantages of B-cell therapy, but how exactly do we actually integrate our constructs into the B-cell? Well, we have an idea for that. Basically what we do is expressing the antibody in one construct, only separated by 2 A sites or by linkers. This creates a vast universe of constructs which you can look up in our basic parts library. We also underlined this with data-driven models which actually prove the correct assembly and binding efficiency of our antibodies. So now it was time to put our elaborate plans into action. First, we successfully cloned over 20 of our constructs, a huge success, which we could confirm with Sanger sequencing. Then we expressed these constructs in XP293 cells. Then we purified them and put them on an SDS page. Here you can see the purification curve, and here the SDS gel. Here you can see how the antibody chains separate under reducing conditions. So how do we prove that these antibodies actually work? For this, we conducted an ELISA with HPV antigen and our antibody. And you can see that our sample has a much higher affinity than the control. We also conducted a hemolysis assay where you apply alpha hemolysin to a blood agar plate and then count how many circles of dead cells you see. And this also showed that our antibodies, if applied with the toxin, protect the cells from death. And although our controls also had some protective effects, so to verify those results, we would have to repeat this experiment again. So, but we did not only produce antibodies, but we also managed to successfully introduce them into Ramos B cells. And this, for us, was huge. So we transfected a linear homology template containing Cas9 2-guard RNAs and truncated EGFR into the B cell. Then we checked for successful transfection with a fluorescent marker on the Cas plasmid, and then we actually sorted for the truncated EGFR to confirm that the construct was successfully integrated into the IDH locus, which was targeted by one of the guide RNAs. The other guide RNA targeted the light chain Ig lambda locus of the B cell to knock out the light chain to prevent endogenous light chain mispairing. So this shows that the B cell therapy could theoretically be applied to all of our constructs and elevates it from a mere antibody expression experiment to a cell therapy of the future.
Hi. And we haven't stopped on the results presented right now, but also are partnering up with stakeholders from both academia and industry to further validate and test our system. On the one hand, we have partnered up with EBD and Brand, 2 companies specialized on cell cultures. With their help, we will be able to test the transcytosis of our antibodies, basically their ability to cross the blood-brain barrier. and validate the results with our AI-powered in silico model. Furthermore, we remain committed to stably introducing our antibodies into the primary B cells, which of course is the therapeutic goal. For this, we partnered up with EBA, who provided us with necessary resources for B cell isolation. Lastly, we are also in active discussion with the Toombs Center for the Toombs Center of Nuclear Medicine, who have developed a unique radioligand, which they are testing with a multitude of different cell therapies. In our case, we would like to partner up to test the ability of in vivo cancer clearance in mice with this institute. However, it's also important to notice that the cell therapies, although really powerful, have limitations that sometimes result in also fatal side reactions. For this reason, we have focused ourselves on safety concept, to introducing a safety concept, with which one of main concerns of the patients who we talked to, Alexander. For this, we have introduced a multi-step safety cascade, starting up with a inducible cusp-based 9 switch. Triggered by a small molecule dimer ether, this will be able to selectively kill the engineered B cells introduced into the body. Furthermore, as you have already talked, our system already includes a TEGFR, the human endothelial growth factor receptor, which can act as a unique tag on the cells to track their proliferation, localization, and differentiation. In addition, it can be targeted by cetuximab for clearance. Furthermore, other, less selective safety strategy may be applied down the line. For example, CD20 overexpression, which can be targeted with the monoclonal antibody rituximab, or even general immunosuppressants to ensure the safety of our project. Finally, we have also partnered up with the Ethics Committee, German National Ethics Committee, and ethical organizations at our institutions to underscore ethical commitment in this project.
By enhancing our safety profile of our B-cell technologies, we actually bring our B-cell therapy closer from bench to bedside. This inspired our team to actually bring. Oh no. How do I get back? Okay, never mind. So to bring our project actually into entrepreneurship. So how do we do that? First of all, we did a big analysis of our landscape by doing a market analysis, a competitor analysis, a SWOT analysis, analyzing. Okay, we don't have that much time. So how do we actually plan to make money? First of all, out-licensing. We try to out-licensing our patents to big pharmaceutical companies that want to elevate their B-cell technologies, making them safer. The other hand, co-development. We want to co-develop B-cell therapies, making B-cell therapies ourselves with emerging antibody therapy producing companies. So how do we actually want to do this? What are our plans? First of all, seed funding. We want to secure seed funding by either venture capital, but also with compliance maps. Then research development, and then IP filing. We want to file IPs at the German patent development department, and also secure, oh wow, it's going way too fast. Yes, we try to, first of all, out-licensing package after IP filing. And then while IP filing, we also are working on Series A funding, which not only will ensure us to not only out-license our package, but also to co-develop current B-cell therapies with our partners. For this, we plan on generating, first of all, preclinical data for pharmacovigilance and also pharmacokinetics. And then we will file an FDA IND submission. Yes. And then after that, we're going to go into preclinical trials, in clinical trials, basically. But what sets us apart from our competitors is that we also thought about our long-term strategy for our business. What do we want to do? We want to file, we want to go public. Why do we want to do that? First of all, this will help us to set us apart, and also to, we are not only focused on venture capitals and have better, and will have better, and we'll be able to make better financial decisions because we're not only dependent on venture capitalists, but also on going public. And we can do that with going to the Hong Kong Stock Exchange, because they actually have they actually have this one rule, this Chapter 18 rule that allows companies and startups that don't have, that have financial records of 2 years and venture capital funding to go public, even though they don't have any like profits yet. So what is our vision? Our vision is to enhance B-cell therapies, making them safer and clearer, and thereby revolutionizing immunotherapy. And we thank, of course, all our partners and the whole team and our advisors. Thank you for listening.